This is the double strand or the double helix of DNA. Make a little nick into this double strand, and then you'll have two single strands. This is a single strand of DNA straightened out so that it's not coiled around. This is a length of nucleotide monomers, and they're connected. This is purple part is called the backbone. Okay, so this is a single strand of DNA. There are 4 nucleotide monomers. Here's how they're connected. Each base is connected to a sugar ring, and that base hangs off one side. The phosphate group of the next base down is what connects two DNA sugar rings. So this is the sugar ring of G, and this is the sugar ring of T, and it's connected with a phosphate group, basically alternating sugar ring, phosphate group, and so on. Finishing all this up, you can see the alternating sugar ring, which are blue, and the phosphate groups. And each nucleotide has its sugar ring and phosphate group. Single strand shows each nucleotide has the sugar ring with a base on one side and a phosphate above it. This has a sugar ring with a base on this one side and the phosphate above it. And what connects the nucleotides are the sugar and phosphates. So this is the backbone of it, and the bases are all hanging off on one side. We can see here the atoms that are added to here, and those sugar rings are all exactly the same. The phosphate groups are all exactly the same. What's different on the DNA single strand are the bases, and that's the only difference. Those are different in their structures, and the structures we'll see are actually kind of similar for these individual bases. Going from the individual nucleotide monomers to the somewhat complicated double helix structure of DNA, shown here, is actually a self-assembly process, starting with the nucleotide monomers. So here's a single strand of nucleotide monomers connected together, T, G, C, A, G, and T. These individual bases require the complementary base to pair with it. So this T requires an A, and this G requires a C. As these bases are called in for the fit, and this one won't work because this is a G with an A, as these bases that require complementary fit are called in, this forms the second strand. So this is a self-assembling polymer, this double-stranded DNA. Each base can form hydrogen bonds, so this A and T is shown with the 2 hydrogen bonds, and the G and C, the base pairing forms these hydrogen bonds, and there are thousands of these nucleotides in the polymer. And so what this means is this is a self-assembling ladder. If you have half of the ladder, you can form the other half. So here's what's shown as the ladder. Here are the ladder rungs. These are the base pairs, A, T, C, G, and so on. And then the sides of the ladder are the phosphate sugar backbone. So this is the deoxyribose sugar ring and the phosphate of one monomer, and then the sugar ring and the phosphate of the next monomer, and so on. And you also have that on the other side.
The parent strand has the letters of those nucleotides, T, G, C, A, G, and T. So this had already formed the A and C base pairs, and then that G wasn't going to work. So instead of the G, this is going to have to be a T, and this will be a G, this will be a C, and this will be an A. And this will represent one strand. This is the parent strand, this is one strand, and then this will be the other strand. It will be the second strand that forms.
This at the bottom here shows the ladder, and this ladder starts to wrap around and curl into a helix, and it's a double helix because there are two strands. This is a single strand. That single strand will naturally form its partner, other strand, with the complementary bases, and then you'll end up with 2 strands. Those 2 strands then coil around and form the double helix 3-dimensional structure. This double helix of DNA, the structure is both simple and complex at the same time. So it's simple in that it's made up of only 4 nucleotides, and it's driven by complementary fit. It would be like having a Lego set that only has 4 parts. And what can you build with only 4 Lego pieces that are the same over and over and over again? The information in the guidebook that codes any individual organism or any individual human is just those same 4 nucleotides. But what makes individuals different, what makes organisms different, is the order of those DNA bases. the specific reading of the A's, T's, C's, and G's is what makes 7 billion humans so different, despite that the nucleotides are exactly the same. The structure of DNA is also complex in that it's a double helix. So you have one strand here that's coiling around, and in the meantime you have a second coil around. That's a coil is called a helix, and you have two of them. So it's a double strand, two strands, and two coils at the same time. So what holds together the two strands are the complementary base pairs hydrogen bonding. So you'd have 2 and 3 and 2 and so on. Those two strands are held together very tightly. It's why DNA material can be preserved and recovered from dinosaurs or fossils. And then the overall structure of the helix, the double helix, is actually held together as well by hydrogen bonding along the length, which is along the vertical. Here's the DNA double helix, and this is not separated into single strands. But what holds these two strands together? Remember, it's these along the length of it. You've got these rungs of the ladder, this on the horizontal. These are the complementary base pairs. So we can see that the shapes are complementary, but what holds those together are hydrogen bonds. This dashed line here, this represents a hydrogen bond. So it's not a covalent bond, it's not an ionic bond, it's a hydrogen bond from the partial negative of this oxygen to the partial positive of the hydrogen. So thymine is this base on the left, and this thymine, is it part of the DNA backbone? Is it attached to a sugar group, or is it attached to the DNA backbone? We know it's not part of the DNA backbone. Remember, the bases hang off one side, but it is attached to a sugar group, also attached to the DNA backbone because the sugar group is part of the DNA backbone. Adenine, that's this base on the right. Hydrogen bond only to thymine? Can it bond to both thymine and guanine, or can it bond to all bases? See here, a little hint is adenine always bonds only to thymine by the shape. It's a complementary shape, and also then held together by those two hydrogen bonds.